Hello बच्चों, let's see the question. So the question says BACs and YACs are, so इन चार ऑप्शन में से हमें वो स्टेटमेंट आइडेंटिफाई करना है जो BAC and YAC को करेक्टली डिफाइन कर रहा है. So the key concept in this question is to understand what is BAC and YAC. So this is a moderately difficult question. So first let's understand what is BAC. So BAC stands for bacterial artificial chromosome. So this is an artificially designed cloning vector. So bacterial artificial chromosomes are designed from a particular plasmid of the bacteria, and it is designed artificially based upon our own needs. So bacterial artificial chromosomes are much better cloning vectors as compared to simply just the plasmids of bacteria. This is because bacterial artificial chromosomes have a carrying capacity of 100 to 300 kb. So this means that we can insert around 100 to 300 kb of gene of our interest. But in case of plasmid, the carrying capacity was less than 10 kb, and therefore the bacterial artificial chromosomes are much more efficient cloning vectors as compared to plasmids. So the bacterial artificial chromosomes have a origin of replication, which is known as the Ori region. They also have multiple cloning sites, which are the sites to which we can add the gene of our interest. They also have selectable markers. And what is very specific in the bacterial artificial chromosome is the F plasmid par region. So this F plasmid, that is the fertility factor which is present in several bacteria, is actually a part of that plasmid is inserted into the bacterial artificial chromosome. So this F plasmid is responsible for keeping a low copy number and to ensure that the entire heavy bacterial artificial chromosome is replicated correctly. So this is the bacterial artificial chromosome, which is used as a cloning vector, which has a much higher capacity than plasmids as the cloning vector. Now the second one is YAC. So YAC stands for yeast artificial chromosomes. So again, the yeast artificial chromosomes have a very high carrying capacity, and they are therefore much but better cloning vectors which have a greater efficiency. So the carrying capacity in yeast artificial chromosome 200 to 500 kb. So this also has a very high efficiency or the carrying capacity. Now, yeast artificial chromosome is also an artificially designed cloning vector which is derived from the chromosomes of yeast. It is genetically engineered to carry the gene of our interest, that is, it is genetically engineered cloning vector. So yeast artificial chromosome have an origin of replication for both bacteria as well as yeast. Then they have some selectable markers. They also have multiple cloning sites for the insertion of the gene of our interest, and they have telomeres at the center of the yeast artificial chromosome, because telomeres are necessary in the stability of the genomic uh, DNA in the eukaryotic cells. So, therefore, telomeres are present in yeast artificial chromosome because yeast is a type of eukaryotic cell. Also, it is known as a shuttle vector, and therefore it has both the origin of replication for bacteria as well as yeast, because this yeast artificial chromosome can be used in both bacteria as well as yeast, and therefore it is known as the shuttle vector. So if we come back to the question, then the first option is that natural DNA obtained from bacteria and yeast. So BAC and YAC are not natural DNA, but they are artificial DNA because we designed that DNA with the help of genetic engineering. So this is not the correct option. So the second option says that useful vectors for eukaryotic gene transfer. So this is the correct statement because we saw that both BAC and YAC have very high carrying capacity, and therefore they are very efficient cloning vectors, and they are particularly used for eukaryotic gene transfer because most of the eukaryotic genes are larger in size, and therefore since both these artificial chromosomes have a higher carrying capacity, they can be used as a vector for transfer of eukaryotic genes. The third option says artificial DNA obtained from bacteria and yeast. So this is also the correct statement because we saw that both bacterial artificial chromosome as well as the yeast artificial chromosome are obtained artificially and they are not natural DNA. So the correct option for this question will be option 4, that is 2 and 3 both. So our final answer for this question is option number 4, that is 2 and 3 both. Hope you understood this well. Best of luck. Thank you.